Hello and welcome to Physionic, where we learn the body from the macro to the micro. If that's something you think you'd be interested in, then consider subscribing. Guys, today's content is another examine piece. This examine piece is going to be covering how your muscles have a sense of memory through their own genetics. So the muscle genetics themselves can remember things from past experiences. So with that said, let's jump into this examine piece. So to give a bit of background on what this paper is trying to accomplish, it's trying to see if we have some sort of genetic modification when we weight train, when we use some form of resistance training. Do we see changes to our genes? And the way this is looked at is by looking at methylation patterns. So using a methyl group, if you've taken chemistry before, it's essentially all you need to know is that it's a tag of some sort that's placed on the sequence of DNA, which then either promotes its activity, its expression, or decreases its expression or activity. So these epigenetic modifications would have impact on our hypertrophic effects in our musculature. How well would they be able to grow or regrow after a period of time of not growing? And on top of that, can those epigenetic modifications be passed on to future generations? So what you do now could have an impact, depending on how this paper turns out, on your future generations if you choose or already have kids. So, real interesting stuff. So let's jump right into the meat of the paper. Let's talk a bit about the experimental approach to this paper. So they had 7 participants, of which they were only able to conclude the study with 6 of them, as one of them had to drop out, but they still got a lot of data from that one individual. And they used a weight training system. So they had a trainer that taught the participants how to weight train. And of course, then they had that trainer stick around for 7 weeks of resistance training with progressive overload as the main principle that they're trying to follow to continue to grow, to have hypertrophy. These individuals were completely untrained before and then went into, of course, training for 7 weeks. Then they had 7 weeks where they reverted back to being untrained in terms of having no exercise for 7 weeks. And then the following 7 weeks, so this is about a 21, 22 week study, then the last 7 weeks were a retraining. So they implemented the same resistance training program, which was 3 times a week for every single week of the training. The first 7 weeks and then the final 7 weeks with again, having that middle 7 weeks being no exercise whatsoever, kind of a, I wouldn't even call it a deload, it's completely unloaded. In terms of their measures, they looked at baseline, so before the training occurred and after training, as well as after every single phase. So the first 7 weeks before and after, then 7 weeks again, looking at after those 7 weeks, and then another 7 weeks, again making that total 21 to 22 weeks of total study time. Now in terms of their measures, they used DEXA scans. So DEXA scanning can tell you how much a muscle might grow. So that's what they used for hypertrophy measures. And they took that at baseline and then they took that after each phase. So the first 7 weeks, then the next 7 weeks, and then the final 7 weeks to determine how much hypertrophy or how much atrophy occurred during each phase. And of course, they couldn't just look at hypertrophy, they had to look at, well, the genes, because that's exactly what they're trying to study. So they had to use a series of different genome analysis. And with that, they looked at epigenetic, they looked at gene expression through mRNA levels, and I'll go into a little bit more, kind of orient you on terms of what that is later on. But with that said, that's essentially all that you need to know for the experimental methods, the experimental approach, and let's jump into the exceedingly exciting results. As usual, I am not going to go over every single figure, but as the paper is linked for you to look at, you can of course check out every single figure, and if you have comments or things that you'd like to mention, then I am always open for hearing those. Now, with that said, we're going to jump into figure 1A. So figure 1A, they are looking at how much hypertrophy and atrophy is occurring over the phases. Do you see hypertrophy in phase 1 with this introduction of this lifting program? Answer is yes, of course you do. So with progressive overload, with an increase in volume, weight lifted in general, then you're going to see hypertrophy. They see that. Now, when you see a cessation of exercise during that middle phase, then of course the unloading phase, you're going to see a decrease in muscle mass. So yes, you see yet again a decrease, atrophy, makes sense. And then of course when you reintroduce the stimulus that you had before, then you see hypertrophy yet again. It's remarkable how the body can adapt, but over those 7 weeks, 7 weeks, and 7 weeks, then you see these predictable changes with a weightlifting program. Figure 1B shows the loading, so in terms of how much weight they've lifted over courses of the 7 weeks, and they have a section for the first loading phase and then the reloading phase. And that's that. That's really as simple as that. They just wanted to show that they are progressively increasing as you would dictate by a progressive overload system. Now, moving on to figure 1C is probably one of the most interesting graphs of the entire paper. It really sets up the story for the entire paper. And without this, we essentially wouldn't get anywhere. And it's a really simple thing. It has nothing to do with genes whatsoever, but that's what the paper is investigating. But we need to know if we're seeing some sort of hypertrophy. And I did mention that we are in figure 1A. We see that we do see an increase in hypertrophy, or we see hypertrophy in general. And that's exactly what 1C shows. So you see the quantification of how much hypertrophy you're getting. Now, in the unloaded phase, which is that second 7 weeks, that middle 7 weeks, you're going to see atrophy. So of course you see the muscle mass then return back to near baseline levels. And then this is what is most interesting. With the reloading phase, where you reintroduce the stimulus of weightlifting, what you find is that not only do you regain the muscle that you initially took 7 weeks to gain, but you gain even more muscle than those initial 7 weeks, which is really fascinating, which means that muscle memory must be playing some sort of factor here. But not only that, your body, your muscles are able to build muscle much, much more quickly than they were in the initial 7 weeks. Because remember, these are untrained individuals, so they've never really undergone any sort of strict programming or lifting routine. So this is all new with that initial 7 weeks. But with the final 7 weeks, this is a reintroduction of that same system, and you're seeing more muscle mass accrued over the exact same time period. That's incredible. And now we are entering figure 2, which is far more genetic and epigenetic based. So figure 2, I'm only going to be focusing on 2A, but you are certainly welcome to look at figure 2B. And essentially what it shows is that you see greater hypomethylation, meaning that you are seeing less methylation of the genes than hypermethylation, where you see increases in methylation. So again, think of methylation as tags that you're placing on the genes to modulate, to change how you see this expression. And generally, not always, but generally you think of less methylation as a greater access to that gene. So having greater levels of potential expression of that gene if you have hypomethylation, and hypermethylation would be decreasing or suppressing that gene's expression. So with loading, you see a certain level of hypomethylation and a certain level of hypermethylation, which is relatively consistent with unloading. So you don't see much of a change. Now, in terms of what those genes are, you would have to look at a few of the other figures. But for just in general, the total number of genes is roughly the same between the initial loading condition and the unloading condition. But again, here we go again with this really interesting change when you see the reintroduction of the lifting routine for those final 7 weeks. The reloading condition, here we see great levels, far greater levels of hypomethylation, meaning that most likely you are seeing far greater potential for more gene expression. And we're going to dive far more into that rabbit hole in just a second. Figure 3 looks all kinds of scary, but don't worry, you don't have to actually interpret a whole lot on this figure. It's essentially just a diagram of a particular pathway, an incredibly well-known pathway that has an effect on all kinds of things. The phosphoinositide 3 kinase AKT pathway, or PKB pathway. Now, this pathway is certainly implicated in cellular energetics, but it's certainly also implicated in hypertrophy. And essentially all they want to show is the different levels of hypomethylation and hypermethylation. Again, hyper being more methylation, more tagging of the genome, essentially suppressing the genome generally, and hypomethylation meaning that less tagging of the genes and therefore greater potential for expression. So the green is a better predictor of hypomethylation, less tagging, and you have red, which is hypermethylation, so decreasing the effectiveness or the expression of those genes. And the intensity of the color is just a rough idea of how much it's swinging towards one direction than another direction. And essentially, this entire diagram just shows you that it's all over the place. There are all kinds of different protein expressions that are being hypermethylated and hypomethylated. It's not like there's just this distinct path of everything is hypomethylated in this pathway, that you're going to get a lot of heterogeneity. Figure 4 is certainly daunting again, but for figure 4a, all they're showing you is what's called a heat map. So, and it's not necessarily related to the temperature of heat, but it's just showing you the different colors of expression. So with green being hypomethylation, so each one of those points, which at the point that you're looking at, well, it's not really going to look like points. It's going to look like a map. So the green is hypomethylation, the red is hypermethylation, added methylation, more tagging, lower expression, I keep saying it because I want to remind you, and the black being essentially no differences. And then they looked at different clusters, and all they're trying to show here is that you're going to have changes in the patterns of expressions or the potential expressions or just the general hypomethylation and hypermethylation based on the clusters of genes that you decide to investigate. So if you look at the differences between like cluster A and cluster B, you're going to see hypomethylation in particular conditions and hypermethylation in other conditions. It's not consistent across the board. So then the next thing that they wanted to see is, do you see a consistent hypomethylation, and does that effectively lead to greater expression? So they looked at that. Figure 4B and C, but I'm just going to focus on B, although it's certainly true for C as well. For figure 4B, you see that with less methylation, hypomethylation, you see greater levels of expression. So that's kind of the general idea when it comes to hypomethylation, that you're going to see increases in the actual expression of those genes. But hypomethylation only means that you have the potential for expression. That does not necessarily mean that that gene is going to be expressed. You need a protein, you need something to attach to that gene, to the promoter section of that gene, and promote the actual expression of that gene into mRNA, messenger RNA. Then the messenger RNA is then decoded into protein, and then from there it can be folded appropriately and put into its particular role and place. So they see with greater hypomethylation, so taking away those tags, that frees up the gene to be expressed, but is it being expressed? And the answer is Yes, there is greater mRNA expression related to greater hypomethylation, or a decrease in hypomethylation. So the less methylation, the more expression. And yes, you actually do see expression as measured by messenger RNA. So figure 5 is another daunting figure, but that's to be expected with a Nature paper. Any Nature paper is going to be extremely heavy and difficult to read through, but we're going to get through it. So figure 5, they looked at just a few different genes that they wanted to see if you see greater expression of those genes and how that relates to methylation patterns or habits in general. And what they find, and I'm just going to focus on the top 2 because those are the most interesting. The top 2, the first one is RPL35A, and that probably doesn't mean anything to you, but if you look up the actual gene, that actually encodes for a protein that is part of a ribosomal subunit. Now, the ribosomes are the part that attach to mRNA and then create polypeptide chains. Polypeptide chains are then folded into the appropriate proteins. So that is incredibly important. It's essentially a marker of protein synthesis. So if you see an increase in these ribosomal subunits, that's a good indication that you're going to see increases in protein synthesis. Now what's interesting here is, again, it's going to follow the same pattern. If you see decreases in methylation, then you typically see greater expression of that gene. But what's really interesting here is that you don't see an increase in the expression of that gene unless you're looking at the reloading condition. Again, it's so interesting around this reloading condition, this final 7 weeks that they looked at when they reintroduced this lifting program. You see all these changes that you don't see with the initial loading condition, the initial 7 weeks. So you see this considerable rise in this expression of this ribosomal subunit. And the other series of graphs is looking at UBR5. UBR5 is a ubiquitin ligase. Now, I'm going to cover a little bit more on that in the next figure because they have an incredibly confusing figure there as well. But a ubiquitin ligase essentially binds a target protein and binds another enzyme, and that enzyme then puts a ubiquitin molecule on your target protein. So what does that mean? Then if you continue to add ubiquitin molecules to that target protein, that essentially sends the cell a signal to degrade that protein. So just like they looked at the protein synthetic ribosome subparticle, they also looked at the opposite. They wanted to look at degradation. Do you see increases in degradation? And based off of the data here, that you do see an increase in degradation, especially around that reloading condition, which is, again, incredibly interesting because you're seeing these changes that you don't necessarily see or not see as many of these changes with the regular loaded condition. Figure 6 is another demonstration kind of figure where essentially they're showing you a bunch of images and they're just showing you that for certain genes you're getting a greater hypomethylation, and for other genes you're looking at greater hypermethylation. So again, hyper being red and hypo being green. They're just showing you diagrams of particular ubiquitin ligases, as well as conjugating enzymes, as well as activating enzymes. And all of these essentially play into the proteolytic pathway, meaning that you are taking target proteins, that can be any protein, and you are ubiquitinating it, you're attaching these molecules to it to lead it to go through proteolysis. So you're leading it to go through degradation. So you're decreasing the protein content within the cell, which is what you would see in atrophy, but it's also what you would see in something like muscle remodeling, trying to get rid of old proteins, trying to get rid of non-functional proteins, and making sure that you're creating more parts for better proteins, more functional proteins. And essentially all they're doing here, and this is the only thing that you have to know, is that they're seeing greater expression of particular enzymes, particular ligases, conjugating enzymes, whatever it might be, and they're seeing decreased expression or potential decreased expression with other ligases. So just different isoforms of particular enzymes that are related to proteolysis are being expressed, differentially expressed. Some are being more expressed, some are being less expressed, or potentially expressed or not expressed. That's it. And the final figure. So the final figure, I'm just going to touch on the first two sections. Again, you're looking at a heat map, but this heat map is specific to a single bout of resistance training. So they didn't want to just look at chronic exposure to resistance training, meaning those 3 times a week for 7 weeks. And then again, of course, you have that cessation, but then you have the reintroduction of those 7 weeks. So in total, they end up resistance training for 14 weeks, just broken up by those 7 weeks. They didn't want to just look at chronic. They also wanted to look at what happens with a single bout, just one exercise session. And fascinatingly, what they find is that they see changes in methylation that are almost identical. They're at least extremely strongly correlated with the same ones that you see 7 weeks later. So chronic and acute see the same methylation patterns. And it also tells you that a single bout of resistance training is already starting to get those cogs rolling towards greater hypomethylation and just priming your nucleus and priming your muscle cells to react to a second bout, a third bout. And in the case of losing that musculature, that you are primed to regain that musculature at a far more rapid rate, already from the very first resistance training bout. Awesome. So let's discuss this a little bit, and I've got several points to make here. What can we conclude from this? What can we get out of this? What are some of the exciting potential conclusions that we can get out of this? Well, the first one is that lifting leads to hypertrophy. I know, I just blew your mind with that one. But with figure 1, we saw that with 7 weeks of progressive overload, that's definitely not the main outcome they were looking for, but they had to show it, and they did show it, and it confirms about 1,000 other studies that have also shown it. Resistance training leads to greater muscle mass, especially if you progressively overload. Shocker. Okay, cool. The second point is that clearly resistance training leads to epigenetic modifications. You're seeing decreases and increases in methylation. And some of those modifications, maybe even many of those modifications, persist after you have stopped resistance training. So if you train for 7 weeks, a lot of those epigenetic modifications maintain through your atrophic state, meaning that when you stop exercise and your muscles shrink back to their basal size, then you still have some of those epigenetic modifications, meaning that it's long-standing, that it will maintain and stay with you for a time. Does that necessarily tell us exactly how long? No, but at least we know that with 7 weeks of training, you can get 7 weeks worth of epigenetic modifications that maintain after the initial 7 weeks of training. So that is really cool. So you get some level of muscle memory that's happening within your muscle cells. They remember what it's like to build muscle, to go through hypertrophy, and are able to differentially favorably, and are most likely favorably going towards modifying themselves so that they are more open and receptive to regrowing that muscle in the future. That is a really, really cool principle. The third point, and feeds into that second point, the second point feeds into the third point, that with reload, you see greater epigenetic modification. So again, you're seeing not only the initial epigenetic modifications, the changes in your genes in terms of how they're expressed, or the potential for them to be expressed, but you're also seeing far greater, thousands of other genes that are being expressed or being liberated to be expressed through this reload. And what we find is that not only are your muscle cells able to hypertrophy back to their original state at an incredible rate, I mean at a fraction of their original rate, but they're able to surpass that level. So 7 weeks, a person who has never trained, have them train for 7 weeks, and then take a clone of them and have them go through the process of retraining again, the clone will regain and surpass in terms of musculature the person who is just first training. So that is some really, really cool physiology, some cellular workings that are happening, some genetic modifications that are happening within your cells that are maintaining over time, but not only just over time, but are preferentially maintaining to a point where later on in your life, at certain points in your life, you could potentially have just a greater capacity just because you lifted earlier in your life. At any point, we don't know the timetables behind this kind of stuff, but it does open the idea that potentially, because you got to a certain size at one point in your life, it's much, much, much easier to reach that point again and to potentially push past that point because you took time off or because of injury or whatever it ends up happening that leads you to stop exercise or stop resistance training for a time. And the final point that I want to make, and probably the coolest point that I want to make here, is that these epigenetic modifications, this much greater hypomethylation that you're seeing with this reloaded condition, is driving increases in protein synthetic machinery, but also driving increases in the expression of ubiquitination machinery or proteolysis, protein breakdown, degradation. And what that means is that potentially, while your cells are perfectly capable of creating proteins and going through the basal protein synthetic machinery on their own, when you initially load them, when you go from a non-exercise state to an exercise state, they have enough machinery most likely to create the protein synthetic material that they need to then induce hypertrophy. But when you take that time off, those epigenetic modifications that remain, when you re-stimulate that musculature, those cells not only remember the old epigenetic modifications, but they introduce new modifications, and those modifications can lead to protein synthesis, which is far accelerated. And the way that they do that is by expressing genes of more ribosomes, creating more protein synthetic machinery to a point where you're not just getting basal levels of protein synthesis, you're getting a far accentuated level of protein synthesis because you have more units, you just have more ribosomes around. So it far quickens the speed by which you're able to achieve that same level. And that would, of course, pan out because that's what we see in the functional measures. When we look at the actual measures of hypertrophy, you see far greater levels of hypertrophy when you reload that condition in the same exact time. So that also pans out when you're looking at ubiquitination. So you're seeing greater remodeling most likely, and this is purely speculation here, but you're seeing increases in ubiquitination enzymes, the E3 ligase, the ubiquitin ligase, which again attaches those ubiquitin molecules to target proteins and has them degraded. That most likely lends to it going through remodeling, trying to make a better, more efficient musculature. So you're seeing both of these massively ramped up in terms of their expression that you didn't necessarily see in the initial loading condition, because the initial loading condition was going through and had enough protein synthesis based off of the stimulus that was given to it. But because your muscle cells remember what it was like, or remember what the conditions were like before, they're able to massively overexpress and get to that point much quicker, and then from there continue that protein synthetic response moving forward. So I mean, that kind of stuff, that kind of memory inside the cells is unbelievable. I mean, people talk about things like steroids and how if you take mass amounts of exogenous testosterone, that leads to more satellite cells, that leads to more hypertrophy. Well, this could lend to the exact same idea. Maybe it is through the exact same mechanism. I don't know that, at least we don't know that yet, but it does show that what happens in the past certainly impacts the future. And because we also know that epigenetic modifications can be passed on to your future generations, it would be incredibly interesting if your exercise patterns, your hypertrophy, your muscular growth now could not only impact your life later on in your life, but also impact your offspring. That would be absolutely mind-blowing. So that's what I've got for you guys. I mean, this paper is incredibly dense. There's tons to it. I certainly recommend that you check it out. And I hope that I did the paper a service. Hopefully you got something out of this. And I certainly hope that I have the immense pleasure to speak with you in the next one. Have a good one, guys. Bye. To a gene, instead of changing the gene itself, is you can modify it. So you can... That doesn't make any sense. Terrible explanation.